Hello, everyone, and welcome to today's live broadcast, QIAseq Digital Sequencing and Biomedical Genomics Workbench, Application to Liquid Biopsy and Bladder Cancer Research, presented by Francesco Lescai, Senior Scientist, Biomedical Genomics, QIAGEN. I'm Alexis Corrales of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by QIAGEN. For more information about our sponsor, please visit www.qiagen.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many of your questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen, or use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Lescai. I will now turn the presentation over to him.
Hi, everybody. Thanks very much for inviting me to give this presentation. I would like to talk about to you today a couple of applications that the whole workflow of QIAGEN allows by connecting what we can offer in the laboratory with what we offer in the bioinformatics and interpretation of the data. So the agenda of today will be going through the application of QIAseq philosophy sample to insight to liquid biopsy. We will look at which solutions we have to enable liquid biopsy applications. We will more specifically look at the QIAseq targeted DNA plugin performance and how you can use the plugin to analyze this type of data. And then we will look at a concrete case of application to bladder cancer data and how we can use the knowledge base in order to classify the variants that we discover in this application. And then we will try to wrap up with some important take-home message. So let's start immediately with looking at how the QIAseq sample to insight approach can be useful for liquid biopsy application. Liquid biopsy, as you know, is an emerging cancer application because it's a non-invasive method to detect tumor-derived mutation and their evolution. And in some cases, it can even be better than the solid biopsy. Here I try to summarize some of these reasons. So if you look at a solid biopsy, this is the classical way of sample a tumor from a patient with a disease. The tumor cells are sampled directly. However, as you know, the tumor is multiclonal in nature. This means that you will only be able to sample the portions of the tumor that you take with the biopsy. On the other side, if you take free circulating DNA from the plasma, for example, here you will have DNA that is contributed from dead cells that are shed into the bloodstream, basically from the whole organism. So tumor-derived alleles will only be a small fraction. This means that it would be very important to detect this very tiny fraction, but at the same time, there is the opportunity to detect and sample the whole genomic variability of the tumor, because then the whole tumor, even what you cannot sample with a solid biopsy, will output in the plasma their DNA. So this means that in order to apply what we know to this great potential of the liquid biopsy, we really need to use a smart approach by combining what molecular biology offers us in the laboratory to a bioinformatics that is designed to leverage this kind of molecular biology innovation. And I will try to show you how in these few next slides. So the first thing that we care about is the connection between all steps of the workflow from the lab from the data generation to the interpretation of the data. And here I'm touching on a couple of products that QIAGEN offers for the sample preparation, like, for example, 2-kit to extract DNA from plasma, circulating nucleic acid, how we can target specific genes that are associated to known diseases using the targeted DNA panels, and then how we can analyze this data by using the Biomedical Genomics Workbench, using in particular the targeted DNA analysis plugin, and then interpret these data using the Ingenuity variant analysis or the QCI interpret. So let's have a look at some of these solutions. First of all, the QIAMP Minilute circulating cell-free DNA kits that have been designed specifically to simplify the concentration and purification of free circulating DNA from plasma. And this kit in particular is designed to yield higher yields and therefore ensure the detection of low frequency biomarkers, because in particular this kit produces more complex library during the sequencing, and therefore it does enhance the capacity of detecting low frequency biomarkers in the data. Then the other product I was talking about is the QIAseq targeted DNA panels. This is particularly important because not only offers a number of solutions to target a specific number of genes that are associated to your disease of interest, but in particular introduces the unique molecular identifiers, the UMI so-called reads. This UMI technology has a number of advantages that I will show in a minute, but just let's have a look in these slides to see how UMI are introduced in the workflow. So these UMI are unique molecular barcodes that are attached to your reads before the target enrichment, the sample indexing, and the amplifications. This allows, when you sequence your data, to identify UMI groups, to map reads according to their position, and then group them according to their barcode. In this way, the UMI allow the generation of consensus reads that corresponds to the original molecule. So your data using UMI will basically correspond to an accurate detection and a higher consensus of the original molecules that you have in the sample before starting the sequencing. This approach has a number of advantages, and in particular, the fact that you can allow for correction of sequencing errors and PCI errors, as this first picture shows. So sequencing error and amplification error happen in two different steps when you prepare your library or when you sequence it, and they end up having the same effect, which is an error in the sequence that you read in your data. Using UMI, you can remove this sort of error during the consolidation, the creation of the consensus by UMI group. And the reason is that if a variant is a true variant that you have in the original molecule, that variant will appear in all reads that have the same UMI barcode. But if that variant has been generated either during the amplification or during the sequencing as an error, then it will not appear in all reads with the same barcode. This allows us to detect that kind of errors and remove this error when you create the consolidated reads. Another important element, which is an advantage of using the UMI technology, is the fact that you can provide a more accurate snapshot of the frequencies of that variant in the original sample. If you look at the bottom group in this picture, you can see again that you have the blue star, which is a true variant, and the red star, which is a sequencing or amplification error. So if you consolidate these reads, you will remove the red star, and you will consolidate all the three reads that have the same barcode, in this case 777. You will also be able to estimate the proportion of that variant in the very original molecules that barcode identifies. So in the correct case, using UMI, you can see that the correct variant has a frequency of 33%, 1 every 3 of the original molecules. But if you don't use UMI, once you have amplified your reads, you will see a completely different proportion, because then the sequencing error will have a frequency of 20% in your read groups. But the other variant, the true variant, will have a frequency of 60%. So this means that in order to really improve the accuracy of the detection and the understanding of the frequencies in the original molecules, you need a representation of the original molecules, and that is what UMIs are designed for. So now we have we jump to the bioinformatics, and it is very important to stress that in order to leverage these advantages that I just showed in the use of advanced technologies in the molecular biology part of your workflow, it is important to have a bioinformatics solution that is able to use that barcode and to perform the analysis by doing what I just showed you, which is understanding how the reads should be grouped and how they map to the original molecules you started from. So the easiest way to do that is using the QIAseq panel plugin in the Biomedical Genomics Workbench. It is very easy to install. Once you have installed the plugin, a number of workflows appear in your Workbench tools, but also a very simple guide that is the one represented in this screenshot here. And this is really the gateway to analyze all your panels that you have targeted using QIAseq digital sequencing products. And the reason is that this guide offers a single entry point into your bioinformatic analysis, and most of the parameters will be pre-configured very easily for you. The user doesn't need to select anything, but is still empowered in modifying all the parameters that wants to modify, if they want to modify the workflow. These workflows in the QIAseq targeted panel plugin have been designed specifically to take advantage of the UMI technologies that I talked about, and to clean the data in a way that improves the accuracy of your results. A very important step in this workflow, which I will only show in highlights, is, for example, the preparation of the reads and the training of the reads. If you look at this picture, and this is something that people working in next generation sequencing know very well, is that a great portion of the data that you collect from a sequencing experiment does not belong to the sample that you're actually interested into. And the reason is that all the technology that we use, the sequencers, they all require a number of adapters, primers, and sequences to tag the samples and barcodes, which means that your sequencing data will contain all these information that needs to be removed in order for you to focus on what is important, which is the DNA of your sample. Removing all this information is not simple, and you definitely need a bioinformatics which is able to do that. The trimming step in the QIAseq panel plugin workflow is able to perform automatic trimming, which is capable to recognize and remove also, for example, the read-through signal, which might happen, for example, when the fragment being sequenced is shorter than the read length, and therefore the sequence might include a read through the reverse complement of the adapter from the start of the other read. The trimming in this workflow is so powerful that it is able to detect this read through even of a single nucleotide. So you understand how accurate your analysis becomes when the removal of this unnecessary information is so accurate in the beginning. And then we have a very important differential filtering cascade in the workflow, which is targeted to filter differentially different type of variants based on their size. And this is obviously meant to improve the precision of your results. Let's have a look at what kind of accuracy we can achieve by using these workflow, because all these characteristics that I just mentioned, they have an effect on your capacity to obtain very accurate results. So in the tests that we have performed, we see very clearly that we reach a sensitivity already at 500 UMI-X of 100% for variants that have a frequency of 5%. And for variants that are more difficult to detect, like those with a allele frequency of 1%, we reach a precision, a sensitivity above 95% at 2500X. And in terms of precision, we reach above 95% in high confidence regions at an UMI coverage as low as 1000X, as you can see in the green picture on the right-hand side. So this is to communicate the high level of accuracy that this workflow allows when performing your analysis. And we have benchmarked our workflow against some of the most known software for somatic analysis. And you can see that our workflow reaches very competitive results in this comparison, with an accuracy of 94% for single nucleotide variants and 91% for insertion deletions. So now I think that we can look a little bit more in detail of how you can use a workflow like this one in application to bladder cancer. So we have performed a pilot analysis, and this is only a pilot because we used 8 samples, and the goal for this analysis was to showcase the workflow that we have from sample to insight. So we collected sample from tissue biopsy, and we extracted DNA from plasma of the same 8 samples using 2 different QIAGEN kits. The tissues were excised macroscopically from the solid tumor, as in a classic solid biopsy, and a high percentage of tumor tissue was expected. And at the same time, all plasma samples were collected within the 24 hours prior to surgical removal of the tumor. So these ensure the accuracy of the comparison. It is also important to notice that the tumor stage was localized. So the tumor was differentiated by was muscle invasive urothelial carcinoma in most of these patients. So the 2 kits perform quite similarly when we look at the recovery, and therefore we decided to go on with the newest kit, the Mini-Elute, which is specifically designed for you to target better the low frequency variants. And in these plots, we consider this measure that we called recovery, which is the percentage of how many variants that have been detected in the tissue can also be detected in the plasma data. And you can see from the picture on the left-hand side of this slide that the recovery of single nucleotide variants is approximately 75% on all samples. And this is a very good result if you consider that the tumor that I mentioned was localized, which reduces the expected levels of variant recovery in the plasma. The recovery of variants for more complex variants instead is a bit more variable. As you can see, we have a lower recovery for the deletions, and we have higher recovery for the insertions. And this is obviously due to the complexity of the variants we are dealing with. Then I think in this application was interesting to look at specifically the genes that have been associated previously with bladder cancer, particularly from our collaborators in Tübingen. So this plot shows a map of the mutations that have been found in each patient in a selected list of genes. Only the genes that have been previously associated to bladder cancer are shown here by row, and we have the samples by columns. Where there is a dot, it means that mutations have been found in the patient. The color of the dot refers to the percentage of how many variants from the tissue has also been found in the plasma, like in the recovery measure of the previous plot. Here you can see that the level of recovery is high for some important genes like TP53, KRAS, HRAS, BRAF. For other genes like TSC1, some cyclins, RB1, which are known to be associated to bladder cancer, they harbor variants only in a few individuals, and we don't have a good level of recovery from the plasma data. This is obviously, as I mentioned, is a very localized tumor, so these might partially explain why we haven't been able to reach a higher level of recovery also for these important genes that normally are associated to the disease. Next, I want to see how we can use our knowledge base in order to empower the interpretation of these kinds of results. So what I've done next is running the variants that we found in our samples through Ingenuity variant analysis. And you can see here in the left-hand side of the slide the workflow that is typical in Ingenuity variant analysis. So for those of you who haven't been using IVA before, IVA has a very powerful workflow system where you can filter sequentially variant according to a number of very powerful criteria. And in this workflow, I can show a workflow filter that is based on the classic known filters to remove common variants. For example, retain those that are predicted as deleterious, retain those that are connected to the biology of the tumor, and additional filters. Here in particular, I have introduced an additional final filter that I called pharmacogenetics, because this is a filter that I designed in order to retain those variants present in genes that are associated to drugs that have been approved in the National Cancer Institute website for bladder cancer treatment. And this is very easy to add to the workflow. As you can see here, I've been adding this step by introducing the right drugs in the list. It is important to notice, by using this very powerful but also very stringent filtering approach, that we still have, after filtering through all these criteria, 5 times more variant in the plasma data compared to the tissue data on the left-hand side. So this big difference is obviously worth investigating, and obviously there is the likelihood that there's a level of false positive which is higher in plasma, because this amount of variant might look rather suspicious. But at the same time, there is the possibility that we are actually doing what we are supposed to do, which is capturing a larger genomic variability than the single biopsy site can be. And of course, further validation are needed, but it's worth investigating further what's present in these variants. And we have done that by using the very powerful approach that is present in the Clinical Insight interpreter. So in this slide, I present a screenshot of the kind of results that you can obtain if you run those variants that I showed in the previous slide through the QCI interpret. So I have obtained the detected variants from the plasma in the Biomedical Workbench, filtered them through IVA, and then used QCI interpret in order to highlight some mutations which are particularly relevant to the disease and classified according to the guidelines by their actionability. It is very easy to do a workflow like this one because the Biomedical Workbench has plugins that allow to connect the Workbench to IVA and also to QCII. And this is crucial because it empowers the interpretation of these variants. And you can see in this slide that although, as I mentioned previously, we cannot rule out the presence of false positives among the higher number of variants that we found in the plasma, it is very intriguing to see that several of these mutations appear to be of interest for bladder cancer and capture that heterogeneity of genomic diversity of the tumor that we know is present in this kind of samples. So if I can wrap up some of these messages, I would like to stress that the QIAseq panel analysis workflow, as I showed in my slides, show a competitive performance, even at allele fractions as low as 1%, and is able to detect single nucleotide variants with a sensitivity of 91.5% and the precision of 96.7. Next, I would like to stress that we've been able to reach a recovery of variants well above 70% for variants that have been identified in the tissue and also called in the plasma. And I would like to stress that obviously a high level of coverage is needed to reduce false positives and to increase the recovery levels, as well as detection of higher tumor genomic variability from the liquid biopsy. And then overall, I hope I managed to convey the message that a coordinated sample-to-insight solution can help streamline the workflow from the library preparation to the variant detection to a sensitivity and a level that is suitable for applications like liquid biopsy. And finally, before taking any of your questions, I would like to thank all the people who contributed to this work, which are across different QIAGEN divisions, and particularly from the Department of Urology of the University of Tübingen, who are really the guys who every day have to manage and help the patients where these data are coming from. Thanks. I'm happy to take any questions.
Thank you, Dr. Lescai, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window, type your question into the box that appears on your screen, and click the Send button. We'll answer as many of your questions as we have time for. So let's get started. Our first question is, how do I access the QIAseq panel plugin?
So this is very easy because when you open up the Biomedical Genomics Workbench, you will have a plugin button on the top right corner of the desktop application. And there you can see which plugins you have activated or the ones that you can download. So it's sufficient to click download on this plugin to install it and have it ready with all the workflows available on your left-hand side as soon as it is installed.
Our next question is, how does the Biomedical Workbench integrate with IVA or QCII?
So this I only briefly mentioned during the presentation. So the Workbench has two plugins that connect directly with the interfaces of IVA and the QCII. So it's really easy to call your variants with a workflow in the Biomedical Genomics Workbench and then send your variants to IVA for further filtering already setting some of the parameters in the filters that I showed in one of my slides, and then also to send your variants to the QCI interpret and get immediately a link with the web page of QCI where you obtain results similar to the screenshot that I showed.
Dr. Lescai, our next question is, what are the factors that might influence most of the detection in plasma of tumor mutations?
This is, I think, a very important question. Thanks for asking it, because I think that the liquid biopsy has really a great potential to making it easier to detect the mutations in the tumor, and also, very importantly, to manage the follow-up of, for example, residual disease that is present after the tumor has been treated. It is very important, however, that the sequencing is carried out with the appropriate coverage, and that of course, your bioinformatics is able to detect accurately those type of mutations. And I hope I conveyed the message that we have a very high level of accuracy. But definitely, it is important that the sequencing coverage is high enough. And obviously, the nature and the characteristics of the tumor influence the detection in the plasma, because the more localized the tumor is, and the more is difficult to detect variants in the plasma, because there will be less output of the tumor into the bloodstream.
What kind of parameters do I need to configure in order to analyze panel data?
So the easy answer would be nothing, because that's exactly what the QIAseq targeted plugin has been designed for, to make it really easy for the user to process data from targeted panel sequencing into the Biomedical Genomics Workbench. So the user will find all key parameters to be already pre-configured according to the panel that they select in the initial guide that I showed in one of my screenshots. So the user really doesn't need to configure anything other than selecting which panel they used for their sequencing. Of course, the user can still decide to change any of the filtering parameters that I showed, for example, when I talked about the differential filtering of the different variants. So the user is still in control of what's happening in the workflow, but it doesn't have to configure anything in order to immediately start analyzing the data.
What level of coverage are recommended to achieve the maximum accuracy?
So this is definitely connected to what I was mentioning before, and as you have seen in our performance data, we show for some of the most difficult application the highest accuracy level when we have a coverage above 2,500x UMI. What is very important to keep in mind is that this coverage will be the coverage after you created a consensus read from all reads carrying the same barcode in the UMI technology. So there is a very nice comparison table in one of the handbooks of the QIAseq panels, and it's very important to highlight that the target mean coverage to reach this kind of UMI coverage, depending on the input DNA in your preparation of the library, might be around 25,000x.
And Dr. Lescai, it looks like we have time for one more question. How can you filter the variants you called in the workbench through Ingenuity variant analysis? So
this is very easy, because using one of the plugins that I mentioned, the IDA plugin, on the plugin you can immediately configure some of the key parameters, like what kind of disease you're looking at, and then the filters in the IDA platform will be set accordingly to the parameters that you've set inside the workbench. The results will be immediately imported back into the Workbench as soon as the analysis in the Ingenuity variant analysis platform has completed. So it's very easy to carry out basic analysis in this way, directly in the Workbench without the need of leaving your desktop application. However, if you want to design more complex filtering, you can click on the link that you receive in the Workbench and access the web interface of IVA, and then add, remove, modify some of the filters in the filtering cascade, like in the boxes that I showed in my presentation.
I would like to once again thank Dr. Lescai for his presentation. I would also like to thank QIAGEN for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through July of 2018. You will receive an email from Labyrinth letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.
Thanks very much. Bye-bye.